Hello and welcome to my channel Omni Bioentrance, and today we will be discussing the TIFR 2025 question paper. I am discussing for the subject biology. So coming to I have discussed only the biology questions. I have not done the other chemistry, physics questions and all, or maths questions, but biochemistry, biology and all the other biology related topics I have answered. So coming to the first question, what is the probability that the second child will be a boy for parents whose first child is also a boy? So here what they have told is the parents are already having a baby boy. Now they have asked, what is the probability that the second child will also be a boy? Now here the probability is just 50% or 0.5% is the answer, because the gender of the previous child, it will not influence the gender of the next child. There are chances that the second child can either be a boy or a girl. So the chances that it is a boy is only 50%, because half. Okay, so because each birth, whether it is the first birth or the second birth, then each birth it is mutually independent events. Then next one, the lesion most commonly induced by UV radiation, we all know it is thymine dimers. Then next one, to estimate the number of fish in a pond, a researcher catches 25 fish randomly, marks them with the yellow paint and then puts them back in the pond. The researcher comes back the next day, again catches 25 fish randomly and finds that 5 of them have yellow paint. So we have to estimate the total number of fish in the pond. So it is given by the formula. This is the catch and mark recapture method. So it has got the formula n is equal to n1 into n2 by r, where capital N is the total population size, n1 is the number of fish initially caught and marked, n2 it is the fish caught in the second sample, and R is number of marked fish recaptured in the second sample. So N1 means fish initially caught and marked. So he has catched 25 fish randomly and marked them with yellow paint. So N1 is equal to 25. N2 is the number of fish caught in the second sample. So 25 fish were caught again the next day. So that is 25, and R is the number of marked fish recaptured in the second sample. So 5 of them had a yellow paint upon second capture. So R is 5. So substitute the values and you will get 125 as the answer. Then next one, in statistics, a distribution is considered symmetric if the mean, median and mode are equal. You get a bell-shaped curve. So if they differ, the distribution is termed asymmetric. Now if the right tail is longer, so the median is going to shift, and if the left tail is longer, again the mean is going to shift. So if the right tail is longer, we have a positively skewed distribution, and if the left tail is longer, we have a negatively skewed distribution. So they have given the data, we have to find the skewness. Now we know that for positively skewed distribution, the mean will be greater than the median, greater than the mode. And if it is negatively skewed, then mean will be less than median, less than mode. Now mean, we know sum of all the values given in the data divided by the number of data. So 2 plus 4 plus 6 plus 6 divided by 4, you get 4.5. Median, you have, it is an even number of data, 1, 2, 3, 4. 4 values are there, so it is even number. So the data we have to arrange either in descending or ascending order. Then you have to take the middle two values that they have given, add them and divide by 2. So that is 5. And mode, if you find the one which is occurring at maximum number of times, so that is 6, which is occurring 2 times, whereas 2 and 4 they are occurring only 1 time. So we find that the mean is less than the median less than mode. So mean less than median less than mode is for negative skewness. So the skewness is negative. Then next one, imagine you are looking at one cancer cell under the microscope, trying to estimate the probability that it will die due to chemotherapy. So you have found out a cancer cell, you are watching that under microscope, and you are estimating the probability that it will die of chemotherapy. You know that approximately 95% of cells treated with chemotherapy turn on the p53 gene. So 95% of the cells which are treated with the chemotherapy, they will turn on the p53 gene, and that these cells have 80% chance of dying. The rest 5% they do not turn on the p53 gene, and they have only 10% chance of dying. What is the probability that the cell you are looking at will die? So the probability that the cell will die is, you have to add up both the probabilities. So probability of the cell dying when p53 is on, plus the probability of the cell dying when the p53 is off. So first we will see the on condition. Here p I have given in bracket, it shows probability. So probability of p53 being on is 95% or 0.95%, and probability of the cell dying when p53 is on is 80%, so 0.8. So the combined probability is, multiply both these, you will get 0.76. Then when p53 is off, they have given p53 being off is 0.05, and percentage dying is 10%, so it is 0.10. Combined probability is this. So the probability that the cell will die is, add up both these probabilities, and you will get it as 76.5%. So coming to the next question, based on the phylogenetic tree provided below, which of the following statement is true? F is more closely related to A than to G. Now we can see here that G has originated or descended from A. So they are more closely related because they share this common ancestor, more recent common ancestor. Whereas F is ancient common ancestor of A and G. It is arising from here. It has bifurcated into B. From B arose A. From A arose G. So F is still far away from A and G. And it is distantly related to A and G. This is the ancient ancestor. Okay. Whereas this is the most common ancestor of A and G. So F is distantly related to both A and G, not closely related. Then coming to the next one, E and D share a common ancestor who is more recent than C and D. Okay. Now the lineage from this is, we will take this one, we will take as node 1, and it leads to B. Okay, node 1 is splitting and leading to B. So from B, 2 branches are arising. One is this one and the other is this one. So the branch leading to the common ancestor CDE, we will take it as node 2, and this one we will take it as node 3. Okay. Now from node 2, the lineage is split into C and the common ancestor of D and E. Okay. So from node 4, this is the fourth one. The lineage is split into D and E. So this must be fourth one. Okay. Lineage is split into D and E. So the most recent ancestor of E and D, of these two, E and D, is node 4. That is the most recent common ancestor of E and D, that is node 4. And the most recent common ancestor of C and D, these two, C and D, 1 minute, of C and D, it will be node 2, this one. That is the most recent common ancestor of C and D, it is node 2. Okay. Now node 4, we know it has descended from node 2. Okay. And node 4, it represents the more recent point in terms of evolution. That is, it has evolved from node 2. So it is more recent than the ancestor in node 2. So the common ancestor of D and E, that is node 4, is recent than the common ancestor of C and D, that is node 2. That is what they have given in option B. E and D share common ancestor. E and D share common ancestor, that is node 4, and it is more recent than ancestor of C and D, that is node 2. So B is the right answer. Then B is more advanced than the others as it is located on the leftmost branch. See the cladogram, it is, or the phylogenetic tree, it is a diagram that shows evolutionary relationship among organisms. It indicates whether they evolved from a common ancestor or not. It does not say anything about like this is more advanced than this. It doesn't say that. So C is not correct. D is also not correct. So a wild type protein and its mutant E107A and E107D were analyzed using isoelectric focusing. Now, which of the following options are most likely to correspond to lanes A and B in the given figure? So, what is isoelectric point? So, isoelectric point, we know it is the pH at which the molecule carries no net electric charge. It is in Victor ionic form. Now, how is this isoelectric focusing done? So here what you do is you prepare an isoelectric focusing gel using acrylamide, and before polymerizing the acrylamide you will add ampholyte into the mixture, and these ampholytes, they are added so as to create a pH gradient. Then you load the protein or the amino acid sample into the gel, apply the electric field, and the amino acid, it will stop moving when the pH of the surrounding gel equals that of the Pi. So that is the point at which the net charge is 0. Now one more point I will tell you is that the basic amino acids, if the amino acid is or the protein is having more basic amino acids in them, then it will have a higher Pi value, and these proteins, they are usually positively charged. And if it is the case of protein having more acidic amino acids, then they will have lower Pi values and they are negatively charged. So a wild type of protein and its mutants, that is E107A and E107D. Now what is E107A? E means your glutamic acid. It has been mutated to alanine. And E, again glutamic acid has been mutated to aspartic acid at the same position, that is at 107. So these are the mutants. What will happen? You have to identify which is A, which is B. Now here if you see the glutamic acid, it is an acidic amino acid. So it will impart a negative charge to the protein, and the protein will have a lower pi value. Okay. So out of these, lower pi value is for B, that is at 7.3. Okay, so it should be your wild type. Okay, if it is glutamic acid. Okay, now if the glutamic acid is mutated to alanine, alanine mutation means the negative charge has reduced. Alanine is a neutral non-polar amino acid. So when glutamic acid got mutated to alanine, there was a reduction in the negative charge. That means the positive charge on the protein has increased. So increase in positive charge means the it will have higher pi values. So here higher pi value is for A, that is around 8.1. So A should be E107A and your B should be your wild type protein, because B is having a lower pi value than A. So your after mutation the pi value increased. So naturally A should be E107A and B should be your wild type. Now why is it not E107D? Because aspartic acid is also acidic, glutamic acid is also acidic. So upon mutation of glutamic acid to aspartic acid, not much change is going to take place in the pi value. Both are acidic. Okay, so as per me, the answer should be B, but in your option answer key they have given it as A. So you can write it in your comment section whether I am wrong. Okay, give your views. Then next question. The pKa of amino acid group of a Switter ion is 9.6. In a 0.1 molar solution of the Switter ion at pH 9, what percentage of the amino group of the Switter ion is protonated? So here we have to use the Henderson-Hasselbalch equation. So it is the relationship between pH and pKa and the ratio of the protonated and deprotonated forms. So pH is equal to pKa plus log concentration of protonated by concentration of deprotonated by concentration of protonated. Deprotonated form is NH2 and the protonated form is NH3 plus. Here they have asked the percentage of amino group to be protonated. So these are the protonated and deprotonated forms of amino groups. pKa and pH is also given. So substitute the values, but we don't have the value for concentration of protonated and deprotonated forms. So pH minus pKa, we will get it like this. So 10 raised to minus 6 is equal to log NH2 by log NH3. Now take antilogs on both the side and you will get it around 0.251. So 0.251 will correspond to the ratio of the protonated and deprotonated form. Now for simplicity's sake, I am taking X or representing X for the protonated form and Y for the deprotonated form. Now Y Y by X, NH2 by NH3 is equal to 0.251. So I have represented this equation in terms of X and Y. So Y value is equal to 0.251X. Okay. So the total concentration is the sum of protonated and deprotonated form, that is equal to 1. Okay, we will take it as 1. So the sum of protonated and deprotonated forms, we are taking it as 1. Now substitute the value of Y in this equation and you will get it as 1.251x is equal to 1 and x is equal to 1 by 1.251. X is your protonated form. So you got the value of x, multiply by 100 and you will get it as 80%. So coming to the next question, the spine of a neuron is approximately a sphere of diameter 1 micromolar meter. The concentration of K plus within the spine is 150 millimolar. So how many K plus ions are in this sphere? Assume Avogadro number. Choose the option closest to the correct answer. Okay, so we have to calculate the number of K plus ions in this sphere. So number of K plus ion, it is number of moles into Avogadro number, and number of moles is concentration into volume. So they have given it is a sphere, so we know the formula for calculating the volume, that is 4 by 3 pi r cube. So from there we will get volume as 5.236 into 10 raised to minus 12 liters. Now calculate, they have given the concentration, that is 150 millimolar. Convert it into molar, that is moles per liter. This is for calculating the number of moles. So concentration, that is number of moles is equal to concentration into volume. So substitute the values and you will get it as 7.85 into 10 raised to minus 17 moles. So number of ions is equal to N into Avogadro number, that is 5 into 10 raised to 7 ions. Then next one, arrange the metabolites in increasing order of phosphoryl transfer potential. In increasing order of phosphoryl transfer potential. Now out of these options, okay, before that phosphoryl transfer potential, it refers to the compound's ability to donate a phosphate group. Now out of these options given, the pyrophosphate, that is S, it is having the least phosphoryl transfer potential and phosphoenol pyruvate is having the highest. So S should come first and Q should come last. So that is seen only in option C. So S less than P less than R less than Q. So the answer is option C. Then next one, an enzyme has a Vmax of 50 micromole product formed, that is minutes into milligram protein, and a Km of 10 micromolar. Here in the question paper also it was a square instead of of the micro representation. So I am taking it as micromolar, and when I took it as micromolar I got the correct answer also. So when a reaction mixture contains the enzyme and 5 micromolar substrate, which of the following percentage of the maximum velocity will be closest to the initial reaction rate? So we know the Michaelis-Menten equation, V0 is equal to Vmax concentration of S by Km plus concentration of S. So Vmax is 50, Km is 10 and concentration of S is equal to 5. So we have to calculate the V0 by Vmax. So V0 by Vmax, concentration of S by Km plus concentration of S, we will get it as 1 by 3 multiply by 100, you will get the percentage. So the answer is 33%. Then next one, the keto, that is left, this one. This is the keto and this is the enol form of guanine. Now the keto and enol form of cytosine and thymine are also given here. Now which of the following bases will the enol form, that is this one, of guanine will stably base pair. Now if you take this one, OH is here, so it will form 3 hydrogen bonds. This is guanine, it can form 3 hydrogen bonds. Now if you take the case of cytosine, it is H over here, it is OH, it cannot form hydrogen bond with amino of cytosine. Okay, but if it is amino of cytosine and the keto form of guanine, so H here, O here, they can form hydrogen bond. N here, NH over here, so this H can form hydrogen bond with N. NH2 here, O here, so H of this NH2 can form hydrogen bond with O here. So the keto form of guanine can form hydrogen bond with amino form of cytosine, but the question is for enol form, so that is not the answer. So amino form of cytosine, no. Imino form of cytosine, it is H here, OH here, they both cannot form hydrogen bond. Then if you take the keto form of thymine, okay, here it is O, here it is OH. So they both, see this photograph here, O, OH here, this is the guanine enol form, this is keto thymine form. So O and H, they can form hydrogen bond. Now N here, NH here. So NH of thymine and then hydrogen and nitrogen, they can form hydrogen bond. Then O here and NH2 here. So O and this H of NH2, they can form hydrogen bond. Okay. So the enol form of guanine, it will form a stable hydrogen bond with the keto form. So keto form of thymine is correct. Enol form of thymine, OH here, it cannot form hydrogen bond. Then next one, if the backbone nitrogen group in amino acid are modified to NHCH3 group, what will be the allowed regions in the Ramachandran map of the modified protein change? So if you take an amino acid, it is having NH2, then a carbon, then carbon is having a side chain and a hydrogen. I will try to write here, this is the NH2, then you have the carbon, then here it will be a hydrogen, and here you will have the R group, and here it will be your COOH. Okay, so that is the amino acid structure. So the backbone NH2, that is this one. This is the backbone NH2 group of the amino acid. It is modified to NHCH3. So one hydrogen is removed out of these two, and CH3 group is added, or a methyl group is added. So what will happen to the allowed regions of the Ramachandran map of the modified protein? Now before coming to this, you know the Ramachandran map, it is a plot that gives an idea about the allowed and the disallowed regions for the dihedral angles, that is your phi and the psi. Now there are regions in the Ramachandran plot which are disallowed, because they are having sterically conflicting amino acid side groups there in the protein. That is, atoms in the polypeptide chain, they come closer together and they cause steric hindrance, and these will come under the sterically disallowed region of the Ramachandran plot. But there are also other allowed regions, that is where there are no steric clashes between the R groups or any other groups of the amino acids. So they are the regions where you will find your beta plates, sheets and your alpha helices. So they are the allowed region, very less steric hindrance. Now now you have a protein which is normal, that is with NH2 attached to it. No change for the NH2. So this protein, it will have certain allowed phi and the psi angles in the Ramachandran plot, and it will fall in the allowed region of the Ramachandran plot. Now what you do is you take this protein, modify the NH2 group and you add the methyl group. So this methyl group, it is a comparatively bulky group when compared to hydrogen in NH2. Okay. So it is comparatively bulky group and it will increase the steric hindrance, and it will result in unfavorable van der Waals interaction between the neighboring atoms in the polypeptide chain. So what will happen? So in the normal condition, wherever the allowed phi and psi angle combination were present in the original protein, they will become sterically prohibited because of this methyl group attachment to the NH2 group. Now they become sterically prohibited means the allowed region for this particular protein, it will shrink, whereas the disallowed region will expand in the Ramachandran plot for this modified protein. So the answer here is option B, allowed region shrink. Then next one, the time required for replication of E. coli chromosome is 40 minutes. But the bacterium is capable of dividing every 20 minutes. So the bacterium divides into 20 minutes. That means the chromosome also has to get replicated so that it is equally distributed in the daughter cells. Now what is the condition here? E. coli chromosome takes 40 minutes. That is more time to replicate its DNA than for the cell to divide. Okay. Now under the right nutritional condition. This is because DNA replication can initiate in the daughter chromosome before the initial replication is completed. Now how E. coli is facing this is when more nutritional or the right nutritional conditions are available is first before the first round of replication finishes, the E. coli initiates a second round of replication from its ori. Ori is the region where your replication starts. So before the first round of replication is completed, E. coli, even before that, it will initiate the second round of replication. So this means that at the time of cell division, the cell has two partially replicated chromosomes. Okay, and by the time the cell has completed its division, two copies of the genome will be transferred to the daughter cells. And this type of replication, it is called as multi-fork replication or overlapping replication rounds. Okay. So here they have asked how many replication bubbles would you observe if DNA replication initiates one additional time over a single cell division cycle. So if the DNA is replicating like how I have told, multi-fork replication, then how many replication bubbles will you see? That is what they have asked. Okay. So E. coli, we know that the chromosome, it undergoes the bidirectional replication. That is, the replication fork will move in opposite direction from the origin. So it will replicate both the strands of the DNA helix. See here, this is E. coli. It is double stranded. I have not drawn double stranded purposely to avoid confusion. So E. coli, it is replicating. It is double stranded. So two these things you will see here. Okay, this is the ori and this is the replication fork. It is moving like this. Okay. This is bidirectional replication. Now what I told you, even before the first round of replication is completed, E. coli will initiate the second round of replication from the ori C. Now this is the first round of replication. Ori C has already split into two, into the daughter cell, daughter DNA. Okay, now the ori C will again form a replication bubble. See here. This is the ori C. This ori C forming a replication bubble here. This ori C forming a replication bubble here. But this is the first bubble. It has not yet finished completing the DNA replication in E. coli. So even before the first one has not completed the replication, two other replication bubbles have already formed in E. coli. This is to compensate this 40 minutes. So how many, they have asked how many replication bubbles you will observe. So this is the first one, this one. This is the first one, this is the second one, this is the third one. So 3 replication bubbles you will see. But if the question is for replication fork, then you see here 1, 2. Here also 3, 4, 5, 6. 6 replication forks you will find. Then next one. A friend of yours measures the quantity of DNA in grasshopper cells growing in the cell culture. Here the question is for quantity of DNA, not number of chromosomes. Okay, number of chromosome is different from quantity of DNA. Okay, be careful. In grasshopper cells growing in the cell culture, cells examined during the G2 phase of the cell cycle. G2 phase means S phase is over. That is, replication is over. DNA replication has taken place. So G2 phase will have double the DNA content. Quantity of DNA will be double than in G1 phase. Okay. So G2 phase they have calculated as 200 units of DNA. So quantity of DNA is 200 units in G2 phase. So cell DNA replication has taken place. What would be the amount of DNA per cell in grasshopper cell in the anaphase of mitosis? Okay. So this is the single chromosome. In the G1, after S, it has doubled its DNA content, or the chromosome has doubled, and this is continuing to the G2 and the M phase. In the M phase there is a prophase, a metaphase, and then coming to anaphase, where the sister chromatids of this particular chromosome, they will move to the poles. Okay, that is what is taking place. So here, after this division is completed, each cell will have one chromosome. We started with one chromosome. It divided 2 chromosomes. Okay. But DNA content in anaphase. Anaphase cell has not divided completely. Only the chromatids have separated from this homologous chromosome. and they have moved to the poles. So DNA content, it was 200 here, it will remain 200 here also. This is in a single cell. Okay, DNA content is 200 itself, but the number of chromosome has changed. This is 1 here in the G2 phase, it became 2 in the anaphase. Okay, so the DNA content is same, that is D is 200 units. Then next one, a woman is heterozygous for both phenylketonuria mutation and for X-linked hemophilia mutation. She has a child with a phenotypically normal man who is also heterozygous for a phenylketonuria mutation. What is the probability that the child will be affected by both the diseases? So they have given woman is heterozygous for phenylketonuria mutation. That means phenylketonuria, we know that it is autosomal recessive condition. She is heterozygous means she is having a normal allele and a mutated allele. Okay, so she is just a carrier for phenylketonuria. Fine. And since phenylketonuria is homozygous recessive, both the alleles should be mutated for hemophilic condition to be seen in the phenotype. Okay, so that is with regard to phenylketonuria. Now mutation and for X-linked hemophilia mutation. So she is heterozygous for hemophilia also. Now it is a hemophilia, this is a sex-linked recessive disorder. So female, she is having 2 X chromosome. Heterozygous means 1 X chromosome will have the normal condition, and the other X chromosome will have the hemophilic mutation. So she is a carrier for this X-linked hemophilic mutation, and she has a child with a phenotypically normal man who is heterozygous for phenylketonuria mutation. Okay, so man is normal for hemophilic condition, but he is heterozygous for phenylketonuria. So what is the probability that the child is affected by both the diseases? So this is female who is heterozygous for phenylketonuria, and male again heterozygous for phenylketonuria. So for the child to have phenylketonuria, the chance is 1/4. Because capital A, it is dominant condition will be prevailing in the child. He will not show the phenylketonuria disease. But small a, if it comes, it is a phenylketonuria homozygous recessive condition. So chances of developing phenylketonuria in the child is 1/4. Now we will see for the hemophilia condition. Women is heterozygous. So capital H is for normal condition and small h is for the hemophilic condition. So these are the gametes, and XY, that is man who is normal for hemophilia. So these are the gametes that are produced, and you will get X capital H and X capital HY, X capital H small h and X small h y. So the child these 3 conditions, that is the one having capital H, they will be normal. They will not show the disease. Whereas this child, he is going to show the disease. So the chance that this child will be diseased is 1/4. Now will be affected by both the diseases. So this as well as that. So you have to multiply the probability. So 1/4 into 1/4 will give you 1/60. Then next one, carbon dioxide in the blood is mostly transported. Okay, that is what they have asked. So we all know that transport of carbon dioxide, it occurs by three methods. One is dissolved in the plasma, that is around 7 to 10% of the carbon dioxide is transported by dissolving in the plasma. Then bound to hemoglobin, that is in the form of carbaminohemoglobin. You can transport carbon dioxide, and this is around 20 to 23%. And as bicarbonate ions, that is in the around 70%. Okay. Now carbon dioxide produced in the tissues, it enter into this RBC in the blood. It will combine with water, and by the action of carbonic anhydrase, it will form the carbonic acid, H2CO3. This is unstable form carbonic acid, and it will split into HCO3 minus and H plus ions. Okay, this is the carbonic acid, bicarbonate and hydrogen ions. So, it is in the form of bicarbonate ion that the carbon dioxide is transported, mainly around 70% of the times. But in the question they have told in the form of carbonic acid, that is H2CO3. Now H2CO3, it is an intermediate precursor to this HCO3 minus, that is the primary form in which the carbon dioxide is transported in the blood. So this H2CO3 is the precursor of HCO3 minus. Okay, so carbonic acid is correct in the form of carbonic acid. C is correct. Then next one, species S1 and S2 use the same resource. So this is S1, S2, they use the same resource R, and S1 is more efficient than S2 in utilizing this resource. Now there is a predator P that consumes both S1 and S2, but at a different rate. Sometimes it will eat S1, sometimes it will eat S2, sometimes it will eat more S1, sometimes it will eat more S2. It is dependent on the prey to choose which one to eat. So which of the statement is correct? P will always, that itself is wrong, eat S2 because it is an inferior competitor to S1. No, need not be. P they have told at different rates. Both the consumes both preys, but at different rate. So it will always eat S2 is not the right answer. Then S1 will outcompete S2 unless the predator eats more of S1. That is correct, because S1 is utilizing the resource more efficiently than S2. So S1 population will increase naturally, and S1 will outcompete S2 if the predator is absent. Okay, no predator, S1 will outcompete S2. Okay, if predator is present and if the predator is consuming more of S1, then S1 population will reduce even though it is consuming more efficiently the resource R. So it will not outcompete S2. So predator is having some impact on the outcome of the competition between S1 and S2. Okay. So S1 will outcompete S2 unless the predator eats more of S1. So if predator doesn't eat more of S1, then S1 will outcompete S2. If predator eats more of S1, then S1 cannot outcompete S2. Okay, so B is right. P has no impact on the outcome of competition between S1 and S2. Yes. That is wrong. P has impact on the outcome of competition between S1 and S2. Just now I told P If it eats more of S1 than S2, then S1 will not outcompete S2. But if P is not eating S1 or S2, P is absent, then S1 will outcompete S2. So P is having some impact on the competition between S1 and S2. Then P will always preferentially consume S1 because it will be more abundant. Again the word always and preferentially it will consume S1. That is not correct. It will consume both the price but at different rate. So the rest I am doing in the next video. So thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of TIFR, you can go to my channel Omni Bio Entrance and you will find them in the playlist. Thank you.